Okay, right. I've just started to record as well. Right. Let's begin then. In this session, we're looking at biology topic 3, looking at infection and response. So this is a really interesting topic. This topic is about disease, okay? This topic's about disease. And we've all heard of disease before, yeah? We've all got sick before. Now, we've all got sick before because one of this or some other microorganism has entered our body. And it's entered our body and it's caused damage to some cells, which has made us be sick, essentially. So the whole purpose of this topic is to talk about disease. Now, if any of you want to do A-level biology, we go into exceptionally great detail on this. It's a really interesting topic. But for now, let's just focus in on what you need to know. Okay, so just to establish things, everything goes back to disease. Now, diseases are caused by pathogens. So diseases are caused by pathogens. Now, we'll talk about what a pathogen is. Now, if you just see in the image, this is an example of a pathogen. Now, all a pathogen is a really tiny thing which causes harm to your body, which causes disease. So you need to know the definition of what a pathogen is. What is this thing? So the definition of a pathogen is that a pathogen, a pathogen is a microorganism, Pathogen is a microorganism which causes disease. Which causes disease. So this is the definition of a pathogen. A pathogen is a microorganism which causes disease. Now, Angela, what do you think microorganism means?
Like
a really small organism. Yeah, you can't see it from the naked eye. You can only see it from a microscope. So, for example, we might have coronavirus. A virus is a pathogen, okay? This is an example of a virus, okay? It's a microorganism which causes disease. Now, you need to know that there's 4 types of pathogen or 4 types of microorganism which cause disease. Now, most of these names you probably know before. So, an example of a pathogen, we've just said one. It's virus. So a virus is a pathogen. Can you name any other pathogens, Selena? Bacteria, yes. Does anyone know any others? Fungi, yes. And there's a final one that many people don't get, beginning with a P. And this is protist. Protist. So these are your 4 types of pathogens. Virus, bacteria, fungi, protist. These 3 should be fairly straightforward. Virus, bacteria, fungi. The final one that you need to kind of try to remember is protist. And we'll talk about what all of these are. Okay, now before we talk about the different categories of virus, bacteria, fungi, protist, let's just talk about how our body protects us against diseases in the first place. How does our body, what mechanisms are in place before this even enters our body. How do we get this out? So let's talk a bit about that. Does anyone want to chuck any ideas at me? How do we prevent pathogens getting in? Vaccines. However, let's say before that, we haven't had any medicine, we haven't taken any vaccines. How does the body naturally prevent stuff getting in? We have the skin, yes. So that's a really, really big factor. Skin. Okay, so look, we can see that we have our skin. The skin is a physical barrier. It prevents viruses, it prevents bacteria getting in. The skin is a huge barrier. Now let's just talk about the skin. If you feel your skin, is it like completely dry usually? Or is it slightly oily? Slightly oily, right? Now, oily. That is a factor. Because the skin is oily, it prevents any bacteria from sitting on it, basically. It prevents any bacteria reproducing. So the fact that the skin is oily is like a natural antibacterial surface, okay? So essentially that's why. Because if your skin's oily, you've got good bacteria on it, and that basically it doesn't have space for any of the pathogens to sit on there. So essentially your skin, yes, is a physical barrier, prevents anything getting in, and also because it's oily and stuff, it doesn't let any bacteria rest on there for long. So that's the first physical defense, our skin. Angela, can you name any others? I mean your hair, yes. You could say that. So yeah, let's put hair. It's not the one I was looking for, but does anyone know any else? Think about if someone sneezed right next to you. It's your nose. Now your nose prevents pathogens getting into your body. Selena, do you want to give a try as to how does your nose prevent things getting in?
Is it because it has like nose hairs?
Yes. That's one, it's got nose hairs. There's also mucus, which traps any bacteria. If you inhale anything, the hairs and the mucus together trap any bacteria that get in. So yes, that's one way that we prevent pathogens getting in. Another way is that in your droplets, in your eye, if you start tearing up, there's enzymes in your eye. So if you start tearing up here, you've got enzymes in that tear, which prevent and kill any bacteria. So that's another factor. Okay, we've also got something in the stomach. What's in the stomach, Isaac? Stomach acid. And what does that do with bacteria if you eat like something that's infected? Breaks it down and kills any bacteria, yeah? So we've got stomach acid and it destroys any bacteria that enters the stomach. Okay, if you just wanted to add that the skin, because of the oil and stuff, is antimicrobial. Antimicrobial. It's got an antimicrobial surface. Basically, what does that mean? It prevents bacteria from sitting on it. Okay. Oh, we've also got our airways. So what's in the airways? Cilia. And what are the cilia pushing? Mucus, yeah. So mucus. We've got mucus in the throat. Mucus in the throat. Now, what does that do? The mucus traps any bacteria that you inhale. Okay, so they're what we call our physical defenses. These are our physical defenses. Essentially, this is how we prevent pathogens getting in. So, let's now discuss what happens when we get pathogens which have now entered our body. So, let's explore that a bit further. So, let's say that a particular bacteria has gone past all of these defenses, and has now entered your bloodstream. So here, look, here we have a pathogen. So look, this is a pathogen which has now entered your bloodstream. And we want to know what happens. How does your body kill it? So, I mean, I'm not sure if you've done this in before, Selena, but we've had a look at what's in the blood. So we've had a look at red blood cells, white blood cells, platelets, and plasma. You're aware of what we're talking about, right?
Yeah.
Okay. Now, for disease, we've got our white blood cells, which are relevant here. So, the pathogen has entered your body. And when the pathogen has entered the body, it starts releasing toxins. So here we can see that the pathogen is releasing toxins. Now, these toxins are what attract the white blood cell to go towards the pathogen. So let's see what happens here. Okay, before we start, you need to understand that the white blood cell has its nucleus, which looks like this. So the nucleus of white blood cells looks like this. Angela, what can you notice about the nucleus? There's more than one part. It's if we look at the nucleus in an animal cell, it's just a circle, right? It's just one part. In a white blood cell, we can see that we've got different lobes, yeah? That's called multi-lobed. So we've got a multi-lobed nucleus. So we've got a multi-lobed nucleus. Essentially, we've got three parts to it. Well, two or three parts to this nucleus. Does anyone know a benefit of it being multi-lobed instead of it being just one circle? So this would be like a grade 7, 8, 9 question. Why is the nucleus lobed? What do you think, Isaac? Oh, okay, Casey's just joined. Hello, Casey. Hi, okay, so, right, you haven't missed much. We actually started a bit late. So all you've missed is about 15 minutes. So that's fine. So I'll just go over quickly what you've missed just so we're on the same page. You're good to start, yeah? Okay, right. We were talking about disease. So we started off with disease, yeah? Here we've got a picture of a pathogen. And a pathogen is essentially a microorganism, a very, very small organism, which causes disease. So if one of this enters your body, it's what causes disease. So that's what a pathogen is. You need to know this definition. It's a microorganism which causes disease. We then looked at the 4 different types of pathogens. There's virus, there's bacteria, there's fungi, and there's protist. Okay, then we moved on to speaking about how does the body naturally prevent pathogens from getting in. We had a look at a range of stuff that's physical defenses. So we had a look at the skin, stomach acid, mucus, hairs in the nose, enzymes in your stomach, etc. And that's where we're up to. So you're fine with all of that, right? Okay, right. So now we were moving on to discuss what happens when we get a pathogen which has now entered your body. So this pathogen has now entered your bloodstream. So the first thing that happens is that this pathogen starts to release toxins. Here it's releasing toxins. And these toxins are what start attracting the white blood cells. Okay, now we were talking about the white blood cell, and we were talking about the nucleus. And we can see that the nucleus has a multi-lobed. It's got different parts. It's not a single circle, okay? The nucleus is lobed. Now, we were talking about the benefit of it being multi-lobed. So I'll just tell you the benefit, but the benefit of the nucleus being lobed is for flexibility, okay? So the reason why the nucleus is lobed is for flexibility. So I'll explain what that means. So, for example, if you get a pathogen which I don't know, has entered a really, really small place in your body, a really tiny, squeezed kind of location. The fact that the nucleus is lobed means that the white blood cell can squeeze in there. So if you ever get a question, explain why the nucleus of a white blood cell is lobed. It's for flexibility. Okay, right. Hopefully we're all now on the same page. Okay, let's move on. Let's discuss what happens. So we've got a pathogen. Selena, can you remind me of what pathogen meant? So what was the definition? Okay, no worries. Did you write it down?
Yeah.
Okay, it's a microorganism which causes disease. I just wanted to check if you knew it. Okay, anyways, yes. The pathogen has now entered our body. So the white blood cell comes in. And the first thing that the white blood cell does is that it checks out its antigens. So if we just remove all the toxins for now. Now, can you see these little triangle bits here on top of the pathogen? Now, these are called antigens. They're called antigens. Now, antigens are on every single body cell. So all of your body cells have antigens on them. So, you know, your muscle cell, your skin cell, all the cells have these little triangle bits on their membrane. They're called antigens. So the first thing that this white blood cell does, is it checks out its antigens. And it realizes that these antigens are not from the body. Okay, so that's the first thing. So I'll write it out just so you understand it. So number one, what's the first thing that's happening? The first thing that's happening is, so after the pathogen has entered your body, is that the white blood cell recognizes the antigens on the pathogen as non-self. Angela, what does non-self mean?
Like it doesn't belong to the body.
Doesn't belong to the body, yes. So that's the first thing that happens. The white blood cell recognizes the antigens on the pathogen as non-self. So this pathogen has entered your body. Your white blood cell comes along and it realizes that the antigens are not from the body. They don't belong here. So that's the first thing that happens. Okay, let's see what happens next. The next thing that happens is that this white blood cell engulfs the pathogen. It eats it up. So essentially, if we've got our white blood cell with our multi-lobe nucleus here, and we've got our pathogen here, what happens is that the pathogen gets eaten up by the white blood cell. So the white blood cell takes in the pathogen. So look here, we've now got our pathogen, and it puts it around a membrane. So that's the second thing that happens. So number two, we have the white blood cell, which engulfs the pathogen. So that's the second thing that happens. The white blood cell engulfs the pathogen. So what was the first thing? The first thing was recognizing the antigens, which are these things, as being not from the body. The second thing is that the white blood cell then eats up or engulfs this pathogen. So Casey, what do you think the final thing that will happen now? Breaks down, destroys the pathogen. So the third thing that happens is that this white blood cell now releases enzymes into here, which destroys the pathogen. So that's the third thing that happens. So number three, the white blood cell destroys the pathogen. And we can see that happening here. The white blood cell destroys the pathogen. So let's say that the pathogen is now destroyed. Now what you need to understand is it keeps these. It keeps these. Casey, why is it kept? Yeah, so sorry, what are these called? Why is it kept behind? Antigens, yeah? Yes, the white blood cell destroys the pathogen, but it keeps behind the antigens. So look, it's kept behind these. And the final thing that it does is it takes these antigens, this is just extra detail, and it puts it on its surface like this. That's what happens. And we'll discuss why it's done that. So number three, the white blood cell destroys the pathogen, yes. And finally, number four, the white blood cell displays the antigens on its surface. On its surface. Okay, so that's what's happening. So just to recap, we have a pathogen which has entered your body. Yeah, it's gone past all of these defenses. It's now in your bloodstream. Your white blood cell comes along and recognizes these antigens as being not from the body. Then it engulfs the pathogen up. It eats it up, it destroys it, and then finally it displays these antigens on its surface, like this. Okay, that is the first thing that happens after we've got past these physical defenses. And this process is called phagocytosis. It's called phagocytosis. So phagocytosis. That's what this process is called. Okay. Yes, Casey? Yes. So, I mean, they're pretty common sense. The skin, stomach acid, mucus in the throat, etc. Have you got it? So the skin has an antimicrobial surface, if you want to add. Basically, it prevents bacteria staying on the skin. Because you've got oil on the skin and you've got other bacteria on the skin, it prevents pathogens from sitting on there. Or growing on there. Okay, right. So, we've just described what happens after a pathogen enters our body. It looks at it, it eats it up, it destroys it, and it displays the antigens on the surface. Right, we now need to discuss what happens after this. So if we go back to this, and if I show you an image of this, does anyone know what we need to do? So if this pathogen comes into our body again, we can ultimately and quickly destroy it. Vaccine, yeah, but what does the vaccine have inside of it? Yes? Or it's got antibodies, right? So yeah, right. Anyways, let's go back to this. So, we're now here. The white blood cell has eaten the pathogen up and it's displayed the antigens on the surface. Okay, I'm just going to draw that out again here, just so you can understand what's happening. So look, here we have our white blood cell with our multi-lobe nucleus. Selena, what was the purpose of it being multi-lobed? Not quite. So remember, it was multi-lobed because for flexibility, yeah, for flexibility. Because if you think about it, if this white blood cell wants to squeeze into tiny places and the nucleus was like this, what do you think would happen? Would it be able to squeeze in tiny places? No. So the reason why the nucleus is like this is so it can squeeze and be flexible. Okay, so that's why. Okay, right. Let's move on to discussing what happens after we've got the white blood cell engulfing and eating the pathogen up. So we're here now. We've got the antigens which are now being displayed on the surface. Okay, what you need to understand is we get another type of white blood cell which comes in. So here, look, we got another type of white. We've got another type of white blood cell which has now come in. And what this white blood cell does is this white blood cell has antibodies on its surface. Has everyone heard of antibody before? Yeah, okay. This white blood cell comes in and it's got antibodies on its surface. And what it needs to do is it needs to match the antibody to the antigen. It needs to match it perfectly. So essentially that's what happens. So here now, look, we can see that we've got a perfect match. We've got a perfect match. So I'll just write it out for you. Another white blood cell comes in. So basically we need to match a specific antibody. We need to match a specific antibody to what, Angela? To the antigen. So that's what we need to do, yeah? We need to match the specific antibody to the antigen. So that's what's happened now, okay? So just to walk you through the stages again. We got this pathogen which has entered your body. It's got past all of these phases. It's now in your body. Your white blood cell comes along, eats it up, destroys it, and it displays these antigens onto its surface like this. Okay, it's now displayed them antigens onto the surface. Another white blood cell comes in and it matches its antibody. It matches its antibody to this specific antigen. So that's where we're up to now. And when we've got this match, this is what makes you immune. This is what makes you immune. Does anyone know what immune means? Yeah, resistant to a specific pathogen. Basically, if you get this disease again, if you get this specific disease again, will you get sick the second time round? No, you won't get sick because you're now immune to that disease. So that's what happens. So let's say that this pathogen has now entered your body again. Okay, so look, after all of this has happened, after we've got it matching to the antibody, let's say, I don't know, 6 months later, this thing enters your body again. This thing has entered your body again. Okay, so let's say 6 months later, you got infected again and it's entered your body for the second time round. Now, what happens in the second time round? Well, what happens is your body detects this pathogen. It detects it. It realizes that this guy has come twice. So that white blood cell comes along and it starts shooting these antibodies at it. It starts shooting these antibodies and they bind immediately. They bind immediately. So what am I trying to say? I'm trying to say that if you get infected again, your white blood cells have already remembered the antigen, so it produces antibodies really quickly and binds to all of them. After it's bound to all of them, we have our white blood cell, which comes in and eats this guy up and destroys it. Eats it up and destroys it. So, what do you need to know? You need to know that if you're infected for the second time round, so if the pathogen enters your body for the second time round, we have something called immunological memory. Now, it's a really big phrase, but if you remember it, it's like 2 marks, I'm pretty sure. So look, you can just say that if you're infected again, if this pathogen enters your body for the second time round, you can say that your white blood cells have immunological memory. Now, it's a big phrase. However, all it means, or you can just say that the white blood cell remembers the antigen. You can also say that the white blood cell remembers the antigen. So that is what this word means, immunological memory. I want to emphasize to you all to please try and remember that. Immunological memory. Basically, if you get infected with this pathogen for the second time round, then your white blood cells have already remembered the antigen, so it starts to produce the the antibodies immediately, okay? And we call that immunological memory. If you can't remember it, just say that the white blood cell remembers the antigen the second time round. Okay, it is complicated, but hopefully I've tried to make it pretty simple for you guys. But just to go over it once more. This is a pathogen. Angela, what was a pathogen? That causes disease. It's entered your before it enters your body. Selena, what are the physical defenses that prevent pathogens getting in? Yes? Mucus, yes, so there are just a few. Right. That pathogen has skipped all of them and it's entered. What's the first thing that happens, Isaac, once it's now entered your body? Yes? Or what was the word? Non-self or foreign, yes. So the first thing is it recognizes these antigens, so these triangles on the top, as being non-self, not from the body. That's the first thing. Then what happens, Casey? Engulfs it and destroys it. It's destroyed it now. Let's say it's eaten it up. Yes, it's eaten it up. Don't say eat, say engulf. It's engulfed it. Yes, it's inside. And like Katie said, it destroys it. Yeah, it's destroyed it. Now, Selena, can you remember what it keeps behind after destroying it? So what does it keep behind after we've destroyed the pathogen? So it begins with an A. Antigens, yeah? Many people get confused, so it's fine if you don't remember it right now. They leave behind antigens. Does that make sense? Okay. Right, just a second. Okay, right. Yes, it leaves behind antigens. Okay, so look, here we've got the antigens left behind. What happens after this, Angela? Yes, good. So we've got antibodies on the surface. So look, before it puts it to the surface, then we've got our white blood cell, which matches the antibodies, and here we've got the antibodies. Okay, we were given a specific, well, we said a specific phrase for the second time round, and what was that, Isaac? Immunological memory, yes. Okay, so that was just as a summary of what we've done. I'm not sure where Selena's gone, but I want to draw a graph up, which is a very, very common graph that you get in the exam. If you want to copy this, we have time, and we have, let's just say amount of antibodies, or concentration of antibodies. Okay, so what is happening? Well, let's say we get infected here. That's the first time that we get infected. So we get our antibodies. Actually, let's say, just a second, let's say we get infected here. That's the first time where we get infected. So we have our, first of all, look, the line is flat. Whoops. Yeah, just a second. Look, the line is flat. And then it rises. Now look, you need to make sure that the line is flat here before we start producing the antibodies. Do you know why, Casey? Why is there a delay? Yes, it has to be detected. It has been detected, but there's something else that the mark scheme wants as to why there's a delay. Isaac, do you know? Yes, good. So look, why is there a delay? There's a delay because it takes time for antibodies to produce. So that's why. So it takes time for the antibodies to be produced. So look, that's why you must have a little delay there. Then let's say we're infected again here, number 2, here. So look, pay very close attention to what happens here. So let's say we're infected here again. So what can you notice, Angela? So what can you notice when we're infected second time round?
take first time from that.
Yes. Hi, Selena. I think you got kicked out of the meeting. Okay, no worries. You're back now. Right, you haven't missed anything, really. We were just discussing antibodies. So if you look here, this is the first time that you get infected with a disease. And it takes time to produce antibodies. And then we were just discussing the second time. So this is the second time when you've got infected. Okay, yeah, Angela, I think you were speaking. Just say immunity, or no, actually no. Say that if you're struggling to remember it, just say that the white blood cells remember the antigen, or remember the antibody. Okay, so yeah, basically we were discussing that this is the first time that you've got infected with a disease. And we can see that, look, we've got the line being flat. The line is flat here. It's flat because it takes time to produce specific antibodies. Whereas, if you get infected with that pathogen for the second time round, there's barely any delay. There's no delay. There's no delay. The reason why there's no delay is because of that memory. Also, look at how many antibodies we produce the second time round. We produce a lot of antibodies the second time round, purely because of that immunological memory. Okay, this is a very, very, very common graph that you get in the exam. And in the exam, this is what they'd want you to do. This is what they'd want you to do. They would have drawn it up for the first time you're infected. And they are saying, complete the graph for the second time you're infected. What are they looking for? They're looking for there being literally no delay, or very, very little delay. And they're looking for it being much higher. So that's how you answer that graph question. Make sure your peak is much higher than the first one. Okay, right. So that's what we've looked at. What time did we start, by the way? Does anyone know? 1:45, yeah. Yeah, so Casey, we started later. Okay, that's fine. Okay, right. So let's just recap where we're coming from. We were looking at disease. We described that this was a pathogen. A pathogen is a microorganism which causes disease. We had a look at how your body prevents it getting in, and then we also had a look at the phases of phagocytosis, where the white blood cell recognizes the antigens as not from the body, it engulfs it, it destroys it, and we're here. Okay, here we get it matching with the with the antibody. So the antibody matches perfectly here to the antigen. So here we get antibodies being made. And that's essentially the process. Basically, you engulf it, and then you find the antibodies. And then we moved on to describing it in a graph. So if we look at this graph, you get infected here. You don't produce any antibodies for a lot of time. So if we have a look at this here, look at how much time this is. And you're not producing anything. This is the time that you're sick. So you know if you get ill, yeah, this is the time that you're sick for. So if you get a question, explain why you get sick for a very long time, or what happens, sorry, explain what happens during the time that you're ill. Well, what happens during this time? During this time, we're trying to find this. This is what happens during the time that you're ill. So if you get a question, explain what happened when you're ill, when you get infected, this is what's happening. We are trying to find, your body is trying to find a specific antibody for a specific antigen. So to answer that question, the reason why you get sick for so long is because this process, finding that specific antibody takes time. It takes time. So this process takes time, and that's why you get ill. That's why you get symptoms of being sick. So that's how to answer that question. Okay. Right, I just want to move on just to discussing how we produce, how we essentially get vaccines on the market or get medicines on the market. Now, before we put a random medicine or a random vaccine out there, what do we need to make sure it is, Selena, before we put it out there? It's safe, that's one thing. Do you know the other? If it works. If it works, that's the second thing. Number one, yes, is it safe or not? And number two, does it work? So that is how we get vaccines on the market. Now, let's walk through the whole process of drug delivery, of drug discovery. So how do we find a drug? How do we test it? How do we put it on the market? How do we get it? How do people use it? Okay, the whole journey, essentially. The first thing that we need to do is we need to find this specific medicine. We need to find, so look, now I'm talking about medicines, okay? Finding medicines. So we're not talking about vaccines right now, we're talking about medicines, okay? So make that different. Right now we're focusing on this. We'll come back to that in a minute. So what you want to know is, how do we find medicine? How do we know that paracetamol is good for reducing pain? Well, how is aspirin good? Where do we get these medicines from? How do we discover them? So the first thing is we need to discover medicines. So if you want to put drug discovery. Drug discovery. Okay. Does anyone know where most of our drugs, most of our medicines come from? Plants. Most of your medicines come from plants. So aspirin comes from willow bark, the bark of trees. Medicines come from plants, most of them. So we need to maintain biodiversity, so we have lots of medicines to discover. So yeah, look, we get most medicines from plants. So let's say you're in your garden, and you think that you found a medicine. You think that you found the treatment to a disease. So what's the first thing that any scientist would do? Does anyone know? The first thing that they do is they would test it on cells. They would test the drug on cells. That's the first thing that they do. They test it on cells. Okay, the second thing that they do after they've tested it on cells, does anyone know what's the second thing that they do before it gets to humans? Animals. We need to do animal testing. So you get the medicine, you get the drug, we test it on cells, we test it on cells, then we test it on animals. And then, number 3, we move on to testing it on a small group of humans. So testing, we can say human testing. Human testing. So these are the 3 things that we do before we get to selling this medicine. We test it on cells, we test it on animals, we test it on humans. Let's just describe the testing on humans. Now, when we are testing it on humans, when we are testing, let's say, this medicine on a group of people, we split them into 2 groups. Does anyone know the 2 groups? There's a placebo group. Yes, we get a test group, we got a placebo group. Now, what does placebo mean? Placebo means you get the fake version of the medicine. So placebo is a fake medicine, essentially. So placebo is fake medicine. That's what it is, fake medicine. So I'll give you an example. Let's say we've got medicine tablets, yeah? Here we've got medicine tablets. And inside this, we've got the drug. Inside this, we've got the drug. And inside this, we've got the drug. In these ones, look, we don't have the drug. We don't have any medicine inside of it. So these ones are called placebo. Placebo is you don't give them the medicine. Okay, so look, we divide them into 2 groups. We divide them into placebo and the people who get the medicine. Okay, now what's the point of placebo? So Selena, what do you think? What's the point of giving people a medicine which doesn't actually have the medicine inside of it?
Is it something what they don't know it's
Yes, it doesn't have any side effects, so yeah, maybe. Do you know the other reason why? The main reason?
no bias.
No bias, yeah. So, for example, just as an example, if someone has depression and they go to their doctors and the doctor gives them a fake medicine, they give them this. And after 2 weeks, the patient comes back and he says to the doctor that I'm cured, my depression's gone. What does that show? They weren't depressed, and also eliminates any type of bias. So the reason why we've got the placebo group is, like Selena said, to eliminate bias. Eliminate any kind of bias. And also we've got the placebo group to see the effectiveness. Effectiveness. Basically, we want to see, does the drug work or not? Okay, so that's what we do. Placebo and test group. Right, now, have you guys heard of blind trial, double-blind trial? Yeah? Okay. So I'll just explain it for the people who don't know. So there's 2 types. So when we're doing this human testing, it can either be blind or it can be double-blind. I'll explain what that means. So let's say we've got all of these medicines and we want to give it to these people here. We want to give it to these people here. Now, what happens in a blind trial? Do you know what happens, Casey? Yeah. So look, if we've got a doctor here, in a blind trial, if we've got a blind trial, the doctor doesn't know which patient's getting it. And so did the patient. The patient also don't know. In a double-blind trial, what's the difference, Isaac? So, in a double-blind, the person who's giving this to the doctor also doesn't know. So, in a double-blind, let's say the researcher. So, the researcher who's giving these medicines also does not know which is fake and which is real. So that's the difference. So just a second. Okay, so just to summarize. Right, so just to summarize, just so everyone understands. Let's say we've got the researcher, we've got the doctor. In a blind trial, the researcher knows and the patients or the people do not know. So in a blind trial, the researcher or the doctor knows, but the people don't know. In a double-blind trial, the people don't know and the researcher does not know. So that's the difference in double-blind. Does that make sense to everyone? In double-blind, both of them don't know. In blind, the researcher does know. So that's the difference. Okay, I think if we move on to doing a bit of English now, to finish the session off, that would be good. But what have we talked through today in biology? Well, we've looked at disease. Now, disease are caused by small organisms. Selena, what were the small organisms called? Beginning with a P. Pathogen. What was a pathogen, Casey? Yes, Angela, what are the 4 types of pathogens? Viruses, yes. Okay, right. Let's move on to English.